everybody. Welcome back to the Doctor is In podcast and our special series, What Plants Crave. I'm your host, Dr. Nadia Sabeh. Many of you know my guest today as a cannabis cultivator and scientist who is willing to share data and insights learned from in-house research and operational practices. She's not shy about what she's learned or what she thinks. And that's probably why we get along so well. Yes, today's guest is Allison Justice, founder and CEO of the Hemp Mine in South Carolina, and founder and manager of the Cannabis Research Coalition in partnership with Clemson University. And as many of you know, is a person who wears many other hats. There are about a million things we could and I think want to talk about related to cannabis, horticultural science, greenhouse versus indoor, the industry in general. The list is long. But for today, as maybe a teaser or maybe just because of special interest in this topic, we're going to try to focus on the art and science of drying and curing cannabis, a subject that I know many of our listeners are going to be interested in learning more about. Allison, welcome to our podcast. I'm so excited to have you.
Hello, Nadia. Thank you so much for having me. I'm excited to be here and to be spending some time with you and our listeners today.
Yeah, I, of course, have wanted you on the podcast for a long time, and I'm excited that you are our 20th guest. It just feels like a good way to commemorate where we've come over the last 8 or 9 months. So, before we get into drying and curing cannabis, I just wanted to catch up a little bit. It has been 2.5 years, I think, since we last talked. And actually, you were on my podcast back then when we were talking about COVID, and the world had literally just shut down. It was April of 2020. And so we're kind of, predicting what was going to happen in the industry and in agriculture in general. How have you been since then? How did COVID affect you? I know one of the takeaways that I had from that conversation was that you were going to be growing some other agricultural crops. I remember okra sticking in my head. Yeah, so tell us what you've been up to.
Yeah, so we did grow quite a bit of vegetables in 2020, and that was fun. That was also hard as a farmer. Speaking of okra, it grows very fast, and if you're not cutting it every single day, it gets very stringy. And so having a team. Yes, so having a team on it to cut every single morning is imperative. But that was good. It was well timed for that year specifically. If nothing else, people always have to have food, and so having local food, where they could come pick it up right on our farm was great. We did only do that for one year because the other thing that people need when they're at home and bored is cannabis products. So, year 2, though, we really enjoyed the experience of getting into vegetable crops. I grew up on a farm. That was nothing completely new for us, but, kind of have to prioritize things. And so we backed off on it after that year. But yeah, it was great. It was a really nice learning lesson. But yeah, no, people enjoy cannabis products too. And, when we spoke back then, the worry was, well, what's going to happen is just everything going to shut down. And, for a while, our retail shops did decrease in orders, but as far as our online sales and that sort of thing, it kind of equaled each other out. When one went down, one went up, and then now it's almost back to normal. So it's not been too bad on us as far as business.
Speaking of hemp, around the same time, there was so much energy and excitement around the federal government legalizing hemp. Has it sort of lived up to that hype, to those expectations, or has it been maybe more challenging than expected for various reasons?
It's an interesting industry, but so is marijuana, right?
Yeah.
The trends swing things, and you're stuck trying to guess what's going to be the fad. Is it going to be rosin vapes, or is it going to be Or is everybody going once Skittles at one
Yeah.
season? And it's the same thing in hemp. I think that the difference is back in 2019, everybody wanted to grow hemp, and they did. I'm talking, doctors quitting their practices and growing hemp.
Oh,
Yeah,
wow.
yeah. Everybody wanted to do it, and they did because they could. And what that did was made this huge, huge surplus, specifically in biomass. And, the next year there was an oversupply. And so, those who wanted to get in it for the quick buck are willing to sell for well below what they put into it just because they're wanting to make a little something back. And so then you're fighting prices, you're competing with people who are just trying to get out, which is terrible. So for a lot of people, the past few years in hemp have been terrible, just to be honest with you. There are people who have picked niches, have done well. We're vertical, so I feel like we've got a little asterisk by our business plan. We're not just growing, we're not just selling young plants. We're able to take that margin that usually you're beat down on from seed to sale. Basically, we're able to keep that full margin because we're selling those consumer good products. But in general, we've been slowly waiting for all of this excess biomass to dry up, and it finally seems, the last couple of months that it has, and the demand's going back up. The prices have gone back up a little bit, and in reality, the prices we had before per let's say kilo of crude was ridiculous. I mean, in that first year, if you successfully grew a crop, you could make $1 million an acre. But at that time, nobody wanted to sell it at those prices because they always thought they could get more. So they held it, and then the market crashed. And so, this circle of learning, and
it's kind of like
I said,
I'm sorry, go ahead.
I was just going to say, like I said, it's the people with niches, those who can grow really high quality smokable flower, or those who have a brand they can kind of go on, or those implementing agritourism. Companies like that can and are doing really well, but those who, are going out and just growing 5 acres for oil extraction and think they're just going to sell the biomass, you may go into the negative.
Is it all about CBD right now? Is anyone growing hemp for fiber?
Yeah, and I think this year has been the biggest so far. We actually planted a little bit of fiber just for fun.
It's a different stock, I guess.
Oh man, it's like bamboo. It's ridiculous. The stems are hollow, they're 15 feet high. And obviously there's different varieties, but the ones we planted, way early in the year still aren't flowering. I thought I'd have to chop them after a couple of months because they would flower and ruin all my females of cannabinoid plants. But no, it just keeps growing. It's crazy. But with that said, I do think that's one of the big futures for hemp. In general, fiber and seed production.
I've heard one of the challenges. I'm sorry. I was just going to say, I've heard one of the big challenges is what you do with that those plants right now. Like there's just not processing plants to use that fiber.
That's the big hurdle. I mean, everybody wants to grow it. If you're only getting $300 per acre for corn, of course I'm going to take $400 to grow fiber hemp, and it's probably easier. So that's coming in a big way. I hear about companies being set up in Kansas and Kentucky, and so hopefully that all comes together, because there's a lot of things that will be replaced for that, and it'll be all in a good way, and be more sustainable.
I was just going to say, and probably be better for the environment than only growing corn and soy all the time. Yeah. So finally, I wanted to ask you about the Cannabis Research Coalition that you started with Clemson University. You have been so busy. Tell us about what that and how that came about.
Yeah, so, I got my PhD under Dr. Jim Faust at Clemson, and in grad school, I was working with floriculture crops, like poinsettias, petunias, that sort of thing. Cool enough, all really relevant, because there a lot of them that I focused on were short day plants, and then also there's a lot to it as far as post harvest. So, taking cuttings in Guatemala, cold chilling them, and then shipping them across the U.S. to be stuck, right? All kind of things that have led me to, I guess, really having a passion in post harvest production, because I got to experience a lot of that with cut flowers and with cutting, so clones. And it's kind of all come full circle, because now Dr. Faust and his plant physiology lab has shifted a lot of focus onto hemp, and it's all hemp grown for cannabinoid, truly for smokable flower. So it's 100% relevant to somebody growing, processing marijuana for smokable flower. And so I guess my point of all that was, when I was in grad school, we had something called the Floriculture Research Alliance. And it was farmers funding research at the university level to, one, have a group you can kind of rely on and talk with, people that are doing the same thing as you, but then also funding for research that's farmer immediately practical, not just research that other researchers like to read, that you might read and forget about. It's research that you can immediately implement into your system and help with yields or efficiency, et cetera, et cetera. So in short, farmer funded research, and that research is then shared with the group. We have monthly podcasts, we'll have an annual meetup, access to certain journals and things like that. So this is where a lot of what we'll be talking about is coming from today. I've had a lot of these theories since 2016 when I got into this, and I've been able to put a few bullet point asterisks as far as data on it, but commercial research is difficult. And so having Clemson involved to do a lot of that nitty gritty research, the grad students funded by the CRC and that sort of thing has been really big in moving some of these questions along, and imperative in my opinion. So I'm excited of what I'll share. A lot of this research, like I said, is done by his grad students.
That's really awesome. We definitely need more of that university industry collaboration. I would say in controlled environment agriculture in general. One, cannabis, obviously, we know what some of the obstacles have been to do that. But even for some of the vertical farming operations, so much of it is so centered around IP and trade secrets that they also don't necessarily want to work with, publicly funded universities and then publicize the research and the research findings. And I do think that it kind of slows the industry down when you don't have the scientists to help advance that research and everyone is so cloistered and doing it on their own. So I'm really excited that you're getting this started.
No, it's
awesome.
Been great. And it's the sort of thing where, they are going to publish because they're a university and that's what they do, but it could take a year or two for publishment to happen,
getting
for sure, past the edits and the hurdles that the journals put out. So, this is just early access is what I like to call it to what may be published eventually.
Absolutely. All right. Let's talk about drying and curing. I know. So you're going to be at the Cannabis conference. We're both going to be at the Cannabis conference here in a couple weeks. Maybe by the time this is posted, it'll be the week of. And so, as a little taste of what you're going to be talking, maybe it's a deeper dive than what you'll be able to talk about in Vegas. But let's start with the most basic question, in my mind, the most basic question. Why is drying and curing cannabis so important? Why are we doing this in the first place?
So if you made me answer with one word, I would say preservation. But from there, obviously, we can expand a lot, right? I think if we're talking human health, which we probably should because it's a human product, preservation would be basically just removing the water to be safe. Right. So like raisins, jerky, whatever, these products can be put into a state of preservation to where they don't have to be put into the freezer or even a refrigerator. And that is ultimately removing enough water to not have microbial growth. So, step one, removing that water for food safety. Obviously, there's mycotoxin and things like that it don't matter how much water you're going to remove, those toxins are going to still be there. But pending that's not an issue we're talking or we're having, it's just simply water removal. Obviously from there, we've got the visual preservation we want to have. So, keeping it out of light, keeps it frosty. We want it to be either green or maybe you have a purple flower. We don't want it brown. So preserving that color. We've got the tactileness, we can call it. We want it firm and dense, which, a lot of that's due to the way it's grown or variety selection. Also thinking of how it feels, you don't want it too wet or too dry, just due to feel, nothing to do with microbials. I think probably the most important that is going to be last discussed is fragrance. We want something that smells nice. We don't want it to smell like grass, and we don't want it to smell like nothing. We want it to smell like it smelled on the plant. And so, that's simply the takeaways of the goals from drying and curing in a nutshell.
Do we necessarily want it to smell like it did on the plant? Or I'm just asking, here's my question. I'm just thinking about like wine, right? So you have a grape and you might eat it or smell it or something, and it has one taste or smell, but then you ferment it, right? You process it, you post-process it, and it might bring out different flavors and different look and feel. I mean, is it necessarily the preservation of what was on the plant, or maybe an enhancement in some way of what was on the plant?
So this is where it gets tricky, right? We are combining art and science here. I mean, way more than the cultivation part, because a lot of it depends on our nose. And my nose talks to me differently than yours talks to you. And, so it's all opinion. And there's certain things we can agree on. We want it to smell like something, and we don't want it to smell like grass or moldy, but there's little nuances in between. If you think about wine, a lot of those things that sommeliers that they're able to notice, it takes them years to develop.
True.
And so I think it's very similar to that, where, I feel like I'm still working on my nose, and I've been on this for quite a while, and then COVID kicks in and that throws me for more. So.
And then people don't even have a nose anymore. So
exactly. So
then it doesn't matter.
That's an interesting point. With wine, with tobacco, there's fermentation that's happening. And so they're taking whatever was in the plant and changing it a little for the good. And I don't think we can put a stamp of, yes, fermenting is happening, but some of the data, we've been able to collect recently is showing basically ethylene being produced when the water activity is below what microbes could grow at. But oxygen and CO2 is not being manipulated in a way to suggest that, basically the plant is still living, respiration. So is that microbes making the ethylene? I don't know, but we can at least put kind of our stamp saying ethylene is being produced. Let's figure out this next step. And that's one of the big takeaways that we were able to figure out in the last group of tests at Clemson.
I'm so glad you brought up ethylene. I still talk to a lot of growers who don't really understand ethylene, who are new to the concept of ethylene and the ripening senescence death chemical, if I may.
That's a good way to put it. Yeah.
I have a feeling we're going to get to there in a minute, but let me just take the next logical question in my head anyway, which is, so why is drying so hard? Like, I don't know. Why not just zap it?
Right. And so these have been really great lessons learned being in hemp and being able to destroy bigger amounts of biomass because it's, I'm not losing. It's not worth as much as marijuana. it's been really great. I'm part of a graduate committee for a lady at UGA, and that's all her doctoral work is on, is different methods of drying hemp. And everything from looking at the trichomes on a SEM, a scanning electron microscope, at different levels of drying. So by levels, I mean just humidity, temperature. So my point is that hard lessons learned in hemp is that drying should be shaped around what your end product is. So hemp, for example, and this is on parallel to marijuana. If you're growing for just isolate, yes, zap it, dry it as hot and quick as you can. You're just going to isolate. And then if we're thinking CO2 extraction for hemp, well, a lot of those companies will put your flower in an oven beforehand anyways to decarboxylate it, because you have better extraction efficiencies when it's in its non-acidic form. So the kind of the preaching I like to do to growers, because in hemp our margins are so much smaller, you've got to be smarter. Figure out what your end game is and then work your way back from there. Because I've seen people go and spend $100,000, $1 million on drying systems when they were just going to produce isolate. Oh man, you're gonna go bankrupt. So again, understanding your end process and then going backwards is the key. But I think our general talk here is about smokable flower.
I think so. Yeah.
So,
but I'm glad you brought that up, because I'm going to ask you at the end just your thoughts on some of the other strategies for drying cannabis that I've seen come through some of the research. We'll get there at the end.
Okay.
But as an exclamation point or something, as a ... Those are totally two different things, but they make sense for me.
Yeah. So if we're talking smokable flower drying, the reason why it's so critical and not just do it is because we have these other little guys in there called terpenes that give that smell of the plant, or at least part of it. There's other things involved that I feel like we're going to put the ... on, because we haven't quite figured out their role, other secondary metabolites. But terpenes, we can test for, our nose can test for it, and we can figure out what's going on with them. They're very volatile in nature, and so drying too fast, they volatilize and leave. And so, if you think about it, if you smell your terpenes, they're not in the plant anymore. So in your drying room, or down the hallway from your drying room, if they're in your nose, they're not in the plant.
Okay, that is a mind-blowing comment. Because when I think about odors, right, and odor control, it's always the drying room that's the stinkiest. And what you just said is, we don't want the drying room to be stinky. We don't want the air to be stinky, because that's those terpenes volatilizing.
Right. And so we need your engineering mind to help us figure out how to keep that in the plant and not in our nose. And which terpenes do we care about? The ones that will volatilize and are the ones that we will eventually smell, whether it's in a jar or in a bag, or do we care about those ones that stay in the flower because they're heavier, but we will actually taste them when we smoke? And so it's been really wild watching, as we do some of these studies, the different types of terpenes, right? You've got your monos, you got your sesquiterpenes, all classified by their isoprene units. So how many
I have no idea what you're talking about right now.
All right, so in general terms, some are heavier than others, right? And so they're going to leave faster than others.
Okay.
Right. So, if you just want to think about it like water vapor, some of the water vapor, some's heavier than others, so they leave faster than others.
Okay.
And so it's been super interesting to dry at different conditions and see how that ratio changes based off of temperature and humidity.
Interesting. So let's just say in the magical perfect world, I know nobody does this, but every grower wishes they could do this. You grew one cultivar at a time.
Mm-hmm.
Theoretically, then, would you maybe want to dry cultivar A little differently than cultivar B if you're trying to isolate specific terpenes differently from them, or not necessarily?
I'm going to say theoretically, yes, but in research, we need to do that, but keep it simple.
Yeah.
For growers out there, I would not be trying to shape this around variety. I think maybe eventually we'll get to that, but I wouldn't go changing up things quite yet.
Okay. So you keep bringing up temperature and humidity, right? So I love that. But tell me, what are the variables that are important? Specifically, you have been talking for a long time, since you started talking about drying, about this idea of water activity. Kind of like me with VPD, I feel like you are making inroads in the community, and I'm hearing more and more people use the term water activity as
Yes.
opposed to moisture content. So congratulations. I'm going to give you a lot of credit for that, if not all the credit. Tell me about what is water activity? How is it different than moisture content? And what are the variables, other than like these terpenes that you were just describing, that we're trying to manage and control?
Yeah, so if we're talking general water content, and this would be basically what I would say the majority of people are measuring their dryness with right now. You think about the meters that have the two prongs that you shove into wood, they're meant for lumber and that sort of thing. They work okay. They work okay for drying and curing. You can usually get them from $30 to $100, and they measure the total water in that plant material. Water activity, on the other hand, is the water that's available there for microbial growth. And so, if we're talking about something that's meant for human consumption, we want to know if we're putting this bud in a position where botrytis can continue to grow and take over that plant tissue. Whatever spore wise or mycelia wise is there to begin with, changing the water activity is not necessarily going to kill it, but it will at least stop it from growing. And so that is the end goal there, because with other foods, we understand the water activity where most, and it'll vary between bacteria and different types of fungi, and so I think we're getting close to putting that exact number on cannabis, but it's definitely a model to go by. And I think we're getting close, because it depends on, how are we talking about 1 gram? Or are we talking about, a 50-gram bud? Because it'll vary, and it only takes a sample from the outside layer.
I was just going to ask, the water activity, or even the moisture content for that matter, I expect is going to vary from the center of the bud to the exterior surface of that bud. So are you looking for the center to be a certain water activity, or the average of the whole bud to be a certain water activity?
So it's interesting. If you have a shorter drying time versus a longer one, your water activity will be different. So let's say I dry really fast for 3 days, and then I put it in a bucket, and then you pull it out and you check your water activity. Well, when you're in the drying room, the middle of that bud is not going to dry as fast as the outside. So, think about a planet. The middle's wet and the outside is dry. Mm-hmm. But you put it into a bucket with a bunch of other buds of similar, and you use water activity meter, you're going to keep measuring what's on the outside of that bud. And so It's going to seem like it's getting wetter, but it's not.
Because the water is migrating
exactly
out.
It's just homogenization. So, saying I want you to hit, in a perfect world, you wouldn't be using a water activity meter that's doing a calculation that's based off the outside of the bud. It would actually sit in there for, hours on end to be able to calculate what's on the inside. but that's not how they're made. So, it depends if you're a short, fast dryer, or if you dry for a longer time. There's some variance there on what you want your set point to be for as far as water activity. But, again, it's guidelines, it's general, because your buds are going to be different sizes, different densities.
Yeah.
All those variables are thrown at you, but we've got to start somewhere, right?
Yeah. Of course. I mean, it just brings up like so many thoughts and questions. So if you dry cannabis more quickly, let's say 3 days versus 10 days, then I guess tell me if I'm understanding this correctly, that in 3 days, you might want to measure a water activity around the surface of the bud that is lower than the water activity if you did it slower, because you're assuming that there's more moisture in the center of the bud. And so you're kind of using that as a gauge for what it's going to end up being. So if I just use a number from 0 to 100, right? And you said, okay, well, I want a water activity of 0.5, or not 0.5, of 50. 0.5 would be really hard.
Well, no, that's actually right. So the scale goes from 1 to 0.
Yeah.
So 0.5 is on the drier side.
Yeah. Right. Because is 0.6, is that the sort of magic threshold?
That's kind of the consumer goal. Yes.
That's the consumer goal?
Yeah.
Okay. So if you're shooting, 0 to 1, and you're shooting for 0.5 at the end, let's say, then a fast drying time, maybe you want to measure, I don't know, 0.4, because maybe the center is 0.6 and the average would be 0.5. I'm literally just making up numbers. So please, listeners, don't use this as your gauge. But if you had drying more slowly and your goal is 0.5, then maybe you want to read closer to a 0.5, because the water should have sort of equilibrated over a longer period of time.
Exactly. And it's good to be able to wrap your head around that. But if you don't care about daily having to basically destroy a gram or two of your bud, the easiest thing to do is to grind it and then measure it.
Oh, okay.
But you understand.
You just made me go through this whole thought experiment.
But you understand it completely, right? I mean,
okay, good. I passed that test. Okay, so there is a relationship, right, between water activity and humidity. Is that right? Mm-hmm. So does 0.6 equal 60% relative humidity?
Yes. So if you were to put the buds in a bucket and it truly did homogenize throughout, basically the headspace humidity should match it. Exactly. So if it were 60% humidity in there, your buds would be 0.6 water activity.
Okay. So since we're talking about time, and I'm just going to use 3 days versus 10 days. You said 3 days. I'm going to say 10 days. Is one better than the other?
Yeah, I think so. According to our research, there's so many asterisks of different variables, varieties, quantity that's in the room, blah, 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 blah. But anywhere from 7 to 10 days, I think, is the sweet spot. I think you're able to keep the temperatures low enough, keep the deficit to a point to where you're not bringing out the water too quickly, or the terpenes are not able to volatilize. And so, pending more research, I think 7 to 10 days is pretty much the sweet spot.
Do you think there's any value to extending that to 14 days or 20 days? Or are we just now sort of playing with fire a little bit because maybe we haven't removed the moisture fast enough that there might be more biological activity?
No, I think you could extend it, but at that point, I don't know if it would be, I think it almost becomes you're turning the drying room into the curing room, because you're not necessarily drying to remove water, you're drying to move water, to homogenize. And so if your drying room was able to, be, and this is one of your questions, I believe, is my ultimate drying curing space would be that exactly. You would do it
in the same space.
Yeah, if I had enough controls. So, we got to figure out what ethylene's doing, if it even matters that it's there, if we're wanting to remove it, if we're wanting to add some to help with, it's the death gas. So, do we want to actually add some or not remove it per se because it's doing something to make smoking better?
Wouldn't that be interesting if we wanted to actually gas the plants with ethylene to
Right.
improve its ripening, like we would a
Mhm.
banana or a
And
tomato?
even before going into the drying room. Yeah. So
Oh man, that's so
yeah,
cool.
I
I
don't
love
know,
this
but
idea.
theoretically, and until I know any different, yeah, I would have a room which would allow me to dry it at the temperature and humidity I wanted. Um, it would be able to be closed off where I could add or take away certain gases or just maintain a humidity. So it basically just become a big bucket. Um, but you wouldn't have to move it. That would be my preference for
Oh,
sure.
so you said something just a minute ago that again sort of rattled my brain. Drying is removing moisture and curing is moving moisture.
Yeah.
I like that. Because I mean, that is one of my questions, which is why are we curing cannabis? What is curing? Is that what it is? Is it moving moisture from the center to the so it's like equilibrated? Is
So
there
I think I can sign my name off on that one, right? I think we can all agree on that. That's
okay?
such an easy one to be certain of. But there's all sorts of other things that we can steal ideas from wine and tobacco and say, well, we know with other plants removing chlorophyll is a thing. It in tobacco you remove chlorophyll. If think back to the old school pictures of tobacco, all the leaves are yellow when they're drying it, and that's on purpose. They want to remove the chlorophyll because chlorophyll provides a harsh smoke, but our buds are not turning yellow. So are we doing that? I'm not sure, but Mike Alden, grad student at Clemson, he will be testing that in the next couple of months. And so I'm super excited to see what he finds out specifically with the chlorophyll, because, it's a solid theory, but they're not, we're not trying to turn the buds yellow. There's other things like, well, how are we manipulating the carbohydrates? So starches to sugars. If you read through the journals on tobacco, they'll actually add sugar to create a better smoke. So,
Really?
and so it's funny, and this is the first time I'm announcing it publicly, I suppose, and I'm proud of it. We've we both have been in this for a while, and we were trained by traditional science. What's right is right, what's written in the papers is what we go by, and then we get to cannabis, and there's guys doing things, and when I say guys, I mean guys and girls. They're doing things very different, and I'm looking at somebody stripping the plants and just dying because, oh my god, you're removing all the photocells. And my point is, there's a lot of practices done where we don't know why.
Mm-hmm.
And I'd say every couple of weeks almost, I'm seeing the data support those ideas. And so the reason
The
I'm
science
bringing this up,
supporting the art,
the
basically.
science is supporting the art more and more. And just this year specifically, I've seen all sorts of folks jump in from university level doing this research, and they're able to put out statistically relevant journals, and I'm like, oh my god, in short, this is just supporting the art. And so I mean, so much so, and we can talk about this in detail later, but so much so I almost want to put out saying, you told me so. You might not have known, but you told me so. Let's talk about why. All of that to lead to flushing. So,
Yes. Yes.
right. So, flushing is a cultivation practice, and it's been done, and it's been done a lot. And, I really think it's to speed up senescence. And is that going to increase yield? No, maybe not. Are you going to be able to put a data point on that? No, probably not as far as terpenes go or cannabinoids. But when we bring out a line of consumers to taste the difference, right? It's all opinion. You might see the difference there, and that removing certain things from the growing side is lending to a better smoke. So that's kind of where the sugars and carbohydrate changes go. Think about a banana ripening.
Yep.
That's what's happening there. I know some people that won't eat a banana once it's like over 2/3 brown because it's converted mostly into sugars.
And it tastes delicious.
And I love it.
Me too.
So who's to say, that's not what some of these practices have been doing all along.
I mean, these are plants. They all kind of do the same thing. I mean, they evolved the same way. So it completely makes sense. And in terms of flushing, by flushing, so I make sure I understand, flushing is the treatment at the end with water, right? That you're just like flushing out. And I know some growers have said, oh, we're depleting it of nitrogen. That's the reason why. And some have said, yeah, it improves the smokability of the flower. So maybe it's helping convert some of those starches to sugars because you've really driven a stress mechanism to the plant, right? I mean, we're just adding another level of stress to say you're about to die. And so now it's like, okay, I'm really dying. I don't even have nutrients to move through my body anymore. And so it starts that conversion process potentially. Has anyone measured the ethylene content in a cultivation facility that you know of? Because wouldn't that be interesting to see that at that flushing period?
Yeah, that would be interesting. I can't say that we have, but we have measured ethylene in a sealed jar on a bud that was harvested but not dried. And it was getting up to 30 ppm.
What's normal?
So let's see. I think for fruit, if you're over like 0.1 ppm, you start having like manipulation of certain things. It would cause plants to curl, or of course bananas would ripen, things like that. So it's a lot, basically
100 times more. Wow. Ethylene is so interesting. Okay, so you sort of described a little bit about what your perfect room would be. Tell me about temperature and humidity. Obviously, we get asked this question a lot. I hear such a range and variety of temperature and humidity combinations that growers are looking for from beginning to end. Some want to hold it steady the whole time. Some want to start higher and go lower towards the end. What are your thoughts on that environmental management?
As long as within the first couple of days you're getting under 0.8 water activity.
Oh, that's it.
The first couple of days. Yeah.
Okay.
Because really that's where microbials can explode, is 0.8 and above. And after that, you can go a little slower. But to be honest, and we're coming back at it again, the 60-60, it works. We found with 60-60, I wish I could pull up a graph right now, but I guess we can post it. Let's see.
Yeah, that'd be awesome.
Yeah, what we did was, take moisture content one axis, and then days and drying on the other, and then look at different temperatures, humidity, so different VPDs plotted throughout. And the one that was best in this representation, we thought was 59 Fahrenheit, 55 relative humidity. And that was about 10 days drying. Again, it's relative to the person, but that also had the best nose. So it seems it's fast enough to stop, terrible microbial growth, but slow enough to really hang on to those terpenes.
And did you maintain that 60-60 the whole time?
That was maintained.
Kind of at the end? Okay.
That was maintained, yeah. To be on the safer side, and a lot of it really depending on the state you're in and what their testing procedures are for microbials. I would maybe say start at 50 and then bump it back up a little bit.
50.
Humidity.
Okay.
Start at 50, and then you can bump back up to 60. But yeah,
But
I
you
think
don't really want to go above 60%, right? Like that's that sort of threshold.
if we're going for high quality marijuana, no, I wouldn't. If we were
Okay.
talking about hemp, I would not be as strict, I'd say.
Okay.
But yeah, I would say that's the goal. Which that lends me to a question for you.
Okay.
So, Dr. Faust, based off of our nose perception and what I just told you, in short, the 60-60, that equates like a 0.8 VPD.
Yeah.
So is it fair to create a chart for VPD for drying? And how do you think that would change drying by being within that same sector within the chart, just as we would in growing?
Yeah, what's funny is I was going to ask you this sort of the same question. Is it VPD that we should be looking at? Because, I plug 60-60 in and I get a VPD of, at least according to our calculator, 0.71 kilopascals. So maybe 0.7 to 0.8 is that sort of sweet spot. And maybe we just say 0.75 for all intents and purposes. Yeah, maybe. I'm sort of curious about that. And I think that's also what lends me to asking the question about temperature and humidity. You're talking a lot about humidity and 50%, 60%, but how does that relate to temperature? I mean, could it be 100 degrees and 50% relative humidity? We know that glass of air is a lot bigger at 100 degrees than it is at 60 degrees. So conceptually, I feel like that's wrong. But if 0.6 water activity is 60% relative humidity, why not have a higher temperature? Is there something else at play? For instance, the vaporization of those trichomes. Is it, if VPD is driving the movement of moisture, is temperature driving something else? The ripening phase, right? The rate of ripening and ethylene production and trichome vaporization. I don't know.
Yeah, I think this is where we get a chemist involved and, start looking at the vaporization of the terpenes.
Yeah.
Because they're all going to be different, and how, yeah, it gets complicated. That's a lot of variables.
It is.
Because it's not, we're not talking about stomates opening. This is physically losing water from, where is it coming out of the most?
Right. And terpenes and trichomes are more oil based, right?
Right.
Or so, God, I guess oil and water, do they mix? I don't know, like how much does oil move with water? I feel like they're almost potentially mutually exclusive, but I could be 100% wrong. I'm not a biochemist in any way. So, but one variable that's not at play with drying and curing is light, right? And photosynthesis and CO2. Well, you tell me about CO2. I shouldn't go there. But when I just think about the photosynthesis curves, right, and finding that optimization point between light and carbon dioxide, and usually it's temperature, though I'm suspicious it's really VPD that we're after, or it's VPD and temperature, because temperature helps drive, right, that metabolism and the rate of photosynthesis. But don't forget about VPD and transpiration, or else you're not moving nutrients through the plant.
Right.
So when I think about drying, there's different variables at play.
Well, I think we can all agree we don't want light in there.
We can agree on that?
Yeah.
Okay. Why? Why not?
Because it turns the flower brown.
Oh, really?
It does.
Any light source?
I mean, if you're okay,
put you on the spot.
Tricky. We don't want to dry in a greenhouse that has no sort of shade. I've done this with hemp, and
really?
the UV turns it brown, even at really cold temperatures. Yeah.
Interesting. Well, I'm up for the experiment. I'm up for collaborating with you guys to try to figure that question out. I don't know the answer, and I guess you don't either, not yet. So that 24 to 48 hours, you mentioned we're trying to get the water activity down to 0.8. I hear a lot of growers who say that they want to remove 50%, usually 50% is the number, of the moisture from the buds within the first 24 to 48 hours. And then after that, it's kind of a slow burn. Would you agree with that practice? Does it
Well,
have
when
to
you
get
say
that low?
50% of the water,
Mm-hmm.
are we saying that the plant, for rough numbers, is 70% water, right?
Yeah.
And so are you saying that they want to get it down to 35%? Or
That is a
is it
very
30?
nuanced question and a fair question. I would say that what they're thinking in their head is that they have 1,000 pounds of wet product, and after 24 to 48 hours, if they weighed it all again, it would be 500 pounds. So not quite from 70 to 35%.
I think in general, that's a fair conclusion. Yeah.
Okay. And then after that, it's just however long it takes to dry it down to your target water activity or moisture content, I guess, in the next 7 to 10 days. Well, I guess 7 days at that point. Is there a correlation between water activity and moisture content? Like, can you take a moisture content measurement and make a guesstimate?
Yeah, we've been able to graph that out.
Okay,
But it's going to vary a little bit with variety,
sure.
basically, because of
Okay,
density and the pieces that come with that. Yeah.
okay. The other question that I always have in my mind too, when I ask that question about the 50% moisture content is, are you only drying the buds? Like, did you wet trim? Right. And you have buds on like baking racks, or is this like the whole plant, right? And you have like the
Mhm.
entire branch like hanging from a rack. So when they say 50%, to me, those aren't equal. But maybe they are.
Yeah, that makes it even more tricky. What I always tell people is if they have a drying room where they struggle to get the water out, well, there's a few things you can do. One is cut off water a little earlier, last couple of days of
Mm,
growing. But two is the more leaves or stems you can remove, that's all physically removing water where your system doesn't
true.
have work to do it. So yeah, there'll definitely be a difference. I'm not sure if I've heard of anybody doing this. Basically, what I'm getting at is understanding if the stem's still on, which loses moisture first, the stem
Mm.
or the bud. I can definitely conclude that the leaves dry first because it's you
They're
can
flat and flimsy.
exactly
They're
they
not
lose
very
it,
dense. Yeah.
and then we go back to drying is removing water, but I'm sure there's an affinity for one to lose it first.
Hmm.
That would be an easy one to kind of quantify. So I'm going to
Hmm.
make a note of that.
Awesome. Yay.
You know what? You can't figure out the answers if you don't have the questions first, right?
That is so true.
That's why I love the CRC is because all these questions like, oh yeah, I didn't think about that, but let's figure it out.
And you talk to a grad student who has no experience with hemp or cannabis and coming in with totally new questions and ideas.
Right.
Yeah.
Right.
That you're like, oh,
Different
wow.
perspectives. Exactly.
Exactly. So what is your preference? I mean, if you baking racks, hanging drying racks, something else, I mean, does it matter so much? Coming from my perspective, I think about airflow a lot. I feel like I don't want to blow too much air at the plant because it would dry it out too quickly. But maybe that's your goal. But like, you still want to have a generally well mixed room, right? Like you would in a cultivation space. And I'll just tell you my thought. Baking racks are way harder to move air through than hanging racks. But what are your thoughts?
Yeah, I agree with that. I agree with that completely. My preference as a cultivator is to do somewhere in between individual buds and full plants. If you've got let's say a 3-foot plant, maybe breaking that down into 5 pieces.
Okay.
So you're getting the majority of the large base stem out of there, but there's still some pretty large stems within the 5 or 6 pieces that's left with the buds. Airflow is important, and it's one of those really, it's crucial, but then it's also, it will ruin the plants. If you have hot spots, botrytis will form. If you've got heavy wind spots, you're going to blow the terpenes right out. And I know that sounds silly on a scientific level, but Dr. Faust had an idea because he saw that. He grows our CBD plants because he has to. So he grows the hemp mine plants, and I've bred them to look and smell like weed. You smoke them, you don't feel anything, but that's the goal for a CBD smokable flower growers, for you not to know. And so he grew a batch, and they were beautiful, and then put them in like a conviron chamber to dry, and had a fan in there, and they smelled like nothing. And he was just so discouraged. And then, I have something that I dried in the back, and it smells better than his. And he's like, I don't understand. So he took a jar. It was a big jar. And he put a fan in that jar and shut it, and the fan, like a small computer fan, and just let it run. And 2 weeks later, opened it up, and it was dry flower he put in there, smelled okay as he put it in there. And when he opened it up, it smelled like nothing. So this is one of those, we don't know what in the world's happening. Where did they go? Because, it's a glass jar. It physically has nowhere else to go except to change. I don't know where it went, but I have experienced, if you have a dry room and, say you have just one big fan in the corner, those that are closest to it, they're going to dry out faster, but then they're also not going to smell great. So, we know boundary layers and, all of these things, but we don't understand yet how those terpenes are evaporating out, or is it actually a conversion into something? The jar example makes me, they didn't just sneak out during the night. They had to have been converted to something else.
So opening the jar, you smelled nothing.
Sealed, fan inside, 2 weeks, open the jar, nothing.
I have no idea.
Yeah, it doesn't make sense. So,
wow.
Point being, there's definitely a finesse of too much and not enough air movement.
Mm-hmm. Let's move to curing real quick, and my epiphany that we're moving moisture through the curing process. Is there another reason why we're curing? And I have to ask a question about burping. Why are we burping?
So
like 2 questions in there, I know.
Yeah. So I think there's an overlap between drying and curing, right? Some people are actually doing a little bit of drying while they're curing, and some are doing a little curing while they're drying. And the only thing I can be sure of is the movement of water. So, we've done studies where we've got a bucket, and let me just I can give you the graphs to post, but just for theory's sake, say you pulled your plants down, they're in a bucket, they're at a water activity of 0.65, right? And they've homogenized. So the head space is also 65% humidity. Well, you burp it, and the humidity in the room is 40%. What do you think you're going to do? You're going to lose water.
Yeah.
So, but then I've also done the reverse where, let's say we're at 0.4, homogenized in that bucket, and then in the room it's 90% humidity. Well, you're going to add moisture to that bucket when you burp it.
Right.
So, there's that factor of just the difference between the two. I can say, going back to ethylene is still being produced on a dry bud. It's minimal comparatively, but in other plants, minimal is a lot. 0.1 ppm is a lot. Mm-hmm. So, is burping releasing that ethylene, supplying something to those microbials that we have not yet identified that are fermenting or are doing something else? I'm not sure, Nadia. I'm not sure.
I have 2 questions around that. One is, just based on this idea of ethylene and even any of the other volatiles, right? Volatile organic compounds that the plant might be producing as it's furthers its senescence process or curing process. I've wondered if part of the reason we're burping is to release some of that ethylene, because if the ethylene concentration gets too high, maybe it prevents the flower, right, from continuing to produce ethylene, right? Because the concentration is so high around it that we might be preventing something. Maybe we want to prevent something, but let's just say we're burping to release that ethylene so it continues the curing process.
Exactly.
So, okay, so now the atmosphere around those buckets, right? The ethylene level is high because we just released all this ethylene into the room around it.
Mm-hmm.
One thought that I've had is that a room, is that a time, a space where maybe we want some outside ventilation to help release that ethylene, to set it back into an ambient condition so that the next time we release that ethylene, right, we've created a concentration difference between that headspace in the bucket and what's in the room. And it probably doesn't take many air change rates, right? It's probably very small, but maybe there's some value there. And again, this might be a research question, but I'm just curious your thought on that.
Yeah, no, I think so. And whether it's an air exchange for CO2 because you're curing in your trim room and there's a bunch of people in there.
Yep.
If it's to move out the ethylene that we just dumped out of the bucket, is it something, like in other plants, aldehydes or those stinky compounds,
Yeah.
are we also, just because we haven't got to measure any of them yet, are we also releasing those aldehydes that, we want to go ahead and sweep them out of the room too? No matter, I guess my point is, whether it's ethylene or whether it's another molecule, that's being put out by the buds. Yeah, I think moving it out of there, out of the room it's in, is probably important too.
Okay. I'm glad that that's not a crazy idea, because I feel
No.
like maybe the drying room is a room where we want to contain, but I feel like the curing room might be a place we want to do a little bit of air flushing. And the other thought that came to my mind as you were describing, if the headspace is 60%, but the room is 90%, or vice versa, the room is 40% and the headspace is 60%. How important is it to control the humidity in the environment around those buckets? Are those buckets sort of self-contained when it comes to the environment? Should we have them insulated so that it doesn't matter what the temperature is around them? Though I guess then when we burp it, if it's 110 degrees in the room, they might feel it then. But do you care if the water migrates into the headspace or out of the headspace, or should they be sort of the same target humidity?
So, no, I think they should be the same target, because if you're burping, it's going to go one way or the other. Basically, when you're burping, it's doing the same thing as putting a Boveda pack in your bucket, right? If it's too high moisture, humidity in there, it's going to bring it down, and then vice versa. And so that's the same thing what's happening when you're burping. So in reality, if, let's say your customer prefers a water activity of 0.65. They like it a little more moist, right? With 0.6 being on kind of the other end. Some people will go down to 0.55 if they're in a state where there's crazy microbial regulations. But yeah, in theory, if you're gonna burp and you're using buckets that are not completely air or gas tight, like the 5-gallon buckets, they're not, you would want to match it, because then you burp and you don't lose and you don't gain. You're just moving gases. You're not moving humidity.
Yeah. So if you want a water activity of 0.65, then ideally the HVAC design engineer, should be shooting for targeting a room humidity of 65%.
Yes.
Okay, perfect. And does the temperature matter in the curing process?
I think the colder you can get it, the better. I mean, obviously you've got to be economical with that, but yeah, if you can keep it in the 60s, Sure.
I feel like that is a point in the curing process where you mentioned, we'll come for a full circle here about preservation. When I think about other foods and preserving foods, their shelf life, we usually chill them. We want to sort of hold them in stasis. And so
Right.
we have a cooler environment so that they don't continue ripening and producing that ethylene. I love food. I love agriculture.
The other thing we haven't talked about, and data coming, but other things that they'll put in jerky and basically to lengthen how long they'll stay good is by putting inert gases in the packaging to reduce oxidation.
Yeah.
And so, interestingly enough, a lot of people have been doing that in the hemp industry, and I'm here to tell you it works. It
Really?
works. It whether it's just keeping those terpenes from degrading into something else, oxidizing. Well, I guess that would be the reason. It works. And so in my dream curing area, I would, once I got to the level I want, which I don't know what that level is yet because we're still doing research, that's what we would do, is inject it with gas. Nobody could walk into that room, but just another method to put it into stasis.
Yeah.
Cool and no oxygen.
Yeah. What inert gas?
So
I have one in mind, but I'm not sure it's the right one. So I want to ask you.
Nitrogen or argon?
Okay. I was thinking argon too. Okay. That wasn't far-fetched. And nitrogen, which makes sense when I think, I love that you like post-harvest. That's actually one of, if I wasn't doing controlled environment agriculture production, I'd probably be doing controlled environment agriculture post-harvest. It's fascinating. It's so interesting what they do. And being in California, there's obviously a lot of post-harvest processing here. But I think about CE rooms, controlled environment rooms, where after they've, harvested the peaches and they put it in this big, like concrete bunker, type of room, and they're just stacked in there and they fill it with nitrogen. Stop. Right. And then the temperature is like 40 degrees. Stop. Right. Like we're literally telling them to stop what they were doing until we put them on the truck, and then we gas them with CO2 so they turn orange because they were probably slightly green when they were picked, and hard and cold and cranky probably. And then we load them up in the truck and flush them with ethylene so that they'll ripen, and raise the temperature so that by the time they get to the grocery store shelf, they're a color that aesthetically look right but probably tastes like crap because they didn't have the starch switching to sugar during the actual on the tree growing, whatever. Everybody knows what I'm saying. So yeah, it makes so much sense to be applying these same farming practices that we've been doing for decades to the cannabis industry. And it makes me wonder, if the hemp growers are doing that, they're probably coming from traditional agriculture and applying traditional agriculture practices to another agricultural crop.
Yeah. And for the most part, most varieties have less terpenes to start with. So you're trying to preserve what little's there.
Yeah. Cool. I don't know. Do you have any final advice for growers who are trying to improve their drying and curing practices other than what we've talked about here? I mean, do you have a bullet list, like do this, don't do that? And maybe that's something we can also share. And I'm sure you will speak to.
Yeah, we'll definitely share some things. But it's funny, one of the things you said we would discuss is kind of alternative drying methods.
I didn't even get to that.
Yeah. And so, I find pretty high value in this. States are going buck wild with the amount of acreage they're allowing marijuana producers to grow. So Michigan, for example, Oklahoma, just 10 acres for one farm, and there's hundreds.
Wow.
This year nobody's ever going to be able to smoke that much. So I work with some of these farmers and I ask, well, how are you going to dry all that? are you doing a stagger harvest where you only have to have half the size of the drying room? Are you going to freeze some up? Like what's your plan? And I've had multiple say, well, we don't know yet. And I'm thinking, well,
Wow.
my God, whether you're having to go out and buy, $50 million worth of HVAC because you're going to dry it off for smokable flower, or if you're going to do something creative with, freeze drying sort of process, it's all going to take time to figure out. One, you better hurry up. But I guess my point here is, for as we get larger and larger scale outdoor grows specifically, we're going to have to be smarter with how we're drying. Going back to, all right, well, you're going to process this amount, we'll send it through this sort of post harvest. Are you going to turn some into extract through butane? Okay, we'll freeze it. Well, if you think just in general terms, I'm going to freeze 100 pounds at a time in a conventional freezer, well, the inside of that batch is going to rot because
Oh
your
my
freezer
God.
can't keep up with it. So implementing, again, just stealing from other industries thought processes, you can freeze stuff instantly. And
Yeah.
there's equipment out there that, it's just a conveyor line. They bring the gas out to you, you freeze it, then you put it in the freezer. Then you can stop and think about what you're going to do with all that flour.
And you can stop and think about it. I love that.
Right. And maybe only do a third as smokable flour. But I think really wrapping, as the price just crashes for hemp and for marijuana, really
Yeah.
taking the time to understand your pathways and how to treat it for post harvest based on what your end product is going to be. I've seen it up and down with hemp, people growing 20 acres and having to just leave 3/4 of it in the field because they don't have the
Wow.
capability to handle it in post harvest. So,
Wow.
it's not just about what the science is going on, it's how to be business minded with that much mass.
Yeah. I think that's sage advice, right? I just know your market and know what your end product is that you're going to be selling. In terms of those other drying practices, I've seen freeze drying, oven drying, microwave, assisted hot air drying. I think there's others that are floating out there. And, you've mentioned tobacco. I also just think about grain drying,
Mhm.
and I mean, coffee bean drying. I mean, there's like so many of our products that you might not even realize like need to be dried. Corn, right? To make your corn chips or your tortillas or whatever. I mean, there's a lot of different drying processes out there for different agricultural products. And, this industry is new, not new. And I do think that there's going to be industrial sort of grade technologies, I guess, and
Mhm.
solutions out there like there is for drying wheat, which we've been
Right.
doing for 10,000 years.
Exactly.
Yeah.
Yeah.
And knowing what that, because I would imagine, I don't know, I'm not a corn anything, but I just feel like, how you post process corn on the cob, frozen corn,
Mhm.
right? Versus, I don't know, dried corn that you get in your salad kit versus masa that you make tamales with versus, the corn for your chips are all different post-harvest handling processes.
On very large scale.
Yeah, on very, very large scale. So if we can do it for corn, we can do it for hemp, right?
Oh, for sure.
Oh, I didn't even mention corn for ethanol. I mean,
Yep.
I don't want to leave them out. Awesome. Well, Allison, I am surprisingly out of questions
Well,
for
that's
now.
awesome.
I know I'm going to
Yeah.
get off. I'm going to get off of this and I'm going to think of another question in 5 minutes. So I'll make sure that I ask you when I see you at the cannabis conference, that question I forgot to ask. But I am really looking forward to seeing you in 3 dimensions again. Looking forward to seeing everybody in 3 dimensions again. And yeah, thank you again so much for being on the podcast.
You're very welcome. Thanks for having me.
Awesome. By the way, before I let you go, how can people find you in the future? And also, is there a way for growers to get involved with your work at the CRC?
Yes, absolutely. So to get a hold of me, there's two websites. There's the hempmine.com, the CRC website. We also have, of course, Facebook, Instagram, my personal one, which I do a lot of research kind of from all of the little pieces I have my hands in, Dr. Justice_grows on Instagram. And then, of course, we have a CRC and a Hemp Mine Instagram webpage as well.
Awesome. Awesome. And for our listeners, Allison is going to provide some of the slides that she was referring to, and we're going to post it on our Dr. Greenhouse Academy, as well as how you can reach Dr. Allison Justice and ask her more questions that maybe I didn't think of here today. I'm sure she would be happy to field them. So thanks again, Allison. Thanks everyone for listening. This was The Doctor Is In with Dr. Allison Justice, our guest, and we will talk to you next time. All right. Awesome.
Yay. That was fun.
That was awesome. I'm so happy you're doing this research.
Yeah. It's mind blowing. It's like you figure one little thing out and then you're like, oh crap, but what about blah, blah, blah.
Isn't that how research?
Yeah, yeah.